Today I'm going to talk about the sexual rotation, about how this process of rotation takes place, and I'm going to focus on the cellular process. To start, I'm going to remind you that this structure that you see here, this group of bristles, yeah, are called the sexcoms, and they are located in Drosophila melanogaster. In the pupal stage, particularly, the sexual rotation takes place between 14 to 44 hours after pupation, although in most of your movies you're going to study this process of sexual rotation between 23 to 36 hours after pupation. We're going to study only this small region called the first dorsal segment, and in that region is where it is located this structure called sexcom. And during development, the sexcom rotation takes place, as you watch in the movie, and we want to know what are the cellular processes involved in the sexcom rotation. What is the cellular basis of the sexcom rotation? And to summarize all the things that I'm going to tell you, I want that just this will be the conclusion. So far, our data shows that the current data are best explained by the push model. So what we found right now is that the cells underneath the comb are expanding while the cells above the comb in the blue region are reducing in optical cell area. And this allows us to make three hypotheses. One, that the cells underneath the comb, here, this guy is pushing this row of bristles. In this case, it will be, we don't know what is the process that is producing this pushing, if it's the increase in optical cell area, we don't know if there is cell division or other process. And what happened above the comb is just a passive rearrangement. It's a result of this push. Another mechanism is a pull. So this guy here, we don't know what is the cellular process that is causing the rotation, but we think if this model is right, there will be a passive rearrangement in the region underneath the comb. And another potential model is that both are correct, that there is a push and a pull. And as I said before, based on our current information, we consider that the push mechanism is the one that is correct. And today I'm going to show you that when we study cell dynamics of sexcom of different lengths and different degrees of rotation, all of them show very consistent results. That is that the cell behaviors in this region will be very heterogeneous. There will be a lot of variation, while the region underneath the comb, in this region, will be very homogeneous, in particular the increase in apical cell area. So this seems to be consistent with the push model. And this seems to suggest that here above the comb, there is a passive response. And this is why there is so much variation above the comb. Later, I'm going to show you that we tried to test this hypothesis using laser ablation, making cuts in the tissue to differentiate between the push and the pull model. And I'm going to show you that why they didn't work as well as we want, and that this led us to do computer simulation. Yeah, and this is the current information about the cell dynamics of the sexcom and try to understand the mechanism underlying this movement of cells. Now I'm going to go through the slides and try to explain in more detail each of these topics. What are the cellular processes that are taking place? Why the push model is the most appropriate model, and what information we have, and how we test those hypotheses. So as I said before, we have three potential models: push, pull, and the combination of both. And here we have these silly cartoons to make it much more clear. And what I want that you think is about these different cellular process, yeah? we can have cell proliferation, we can have an increase in apical cell area, we can also have a reduction in apical cell area, and we can also have cell extrusion and exchange of cell neighbors. And I wonder, you think, could the cell proliferation produce the push? Or maybe the cell extrusion could produce the pull? What kind of cellular process do you think that are involved? In addition, do you think that it's only one cellular process, or they could be a combination of cellular processes? And if you see, using these movies, we can test those hypotheses, and we can measure, we can detect if there is cell proliferation, we can detect if the cells are getting bigger, we can see if the cells are mixing, if the cells are dying. And in the following slides, I'm going to show you an example of how we test those hypotheses. So here we have our tree model, and for example, the increase in apical cell area can definitely can be the force that drives the movement of the comb if it's underneath on the comb. So for example, here you can see that these cells definitely, they start getting bigger, they are small and they get bigger during rotation. Another process that we can test is there is cell proliferation, and definitely we can test those things. And also we can test if, like in our process that I will explain in more detail later, they could be the cells are mixing, and this can lead to a reorganization of the region underneath the comb, and those movement can lead to a movement of the comb. On the other hand, for example, you can also see that the cells start getting smaller, and there are several projects that we are studying to understand how the cells are getting smaller, and if this process could lead to is important or not for the rotation. And also, this is one of the most interesting things. Believe it or not, uh, if I ask you how many cells are dying in, how many cells you see dying in this movie, maybe if you don't see any of those cells dying, don't worry. I watched this movie for many years and I didn't see any. And it was an undergrad student that found that actually 30% of that tissue display apoptosis. There are a lot of cells that are disappearing, and we will talk about what are the cellular patterns, the dynamics of this cell death. Cell death, what we wanted to test, if there was a lot of cell death, maybe above the comb, maybe this cell death can be, as there is a gap in the tissue, this can pull the comb, and this also could be a potential hypothesis that we can test. And finally, the last hypothesis is that there could be a combination. Maybe it's not only one, it's not a single process. It could be a combination of processes that are happening at the same time. And I'm going to show you that actually most of these cellular processes are taking place at the same time. What is fascinating, but it make a little bit more complex to answer this question. Yeah. So the methodology, as maybe you have listened to me talking a lot about this, we use this technique called four-dimensional live imaging. So in addition to the 3 dimensions, we add time. So this way is called four-dimensional live imaging. And as I said before, we study between 23 to 36 hours of occupation because it's the time of the morphogenesis of the pupil where the comb move the most. And we took pictures like this ones to quantify spatial temporal patterns. So, for example, one that they were very important is the changes in apical cell area. And we study also type of cell rearrangement. We track cells. We can do, we see if the cells were dying. We can do a lot of things, and I'm going to show you some examples of those. One topic that we will talk in more detail later, but right now I want that you make sure that you remember is the concept of cell polarity. That refers to the spatial difference in shape, structure, and function within a cell, and there is 2 types. the apical and basal cell polarity and the planal cell polarity. This apical and basal cell polarity is very important for us. Why? Because what we image of the cell is this subapical region. Here you will have the cell matrix here. What you are seeing in all these movies is the subapical region. Yeah, so let me show you. This is a diagram, and this is from vertebrates, this is non-vertebrates. Here you have in the cells the apical and basal polarity. You have different proteins, but what I want that you pay attention is that the cells to attach to each other, they have something called adherent junctions, and in the most important, one of the most important one is this decoherent complex that are located in the subapical region, not a completely apical, subapical region, and we put a GFP. We put a GFP in this gene, in this protein, and now what we see in all these movies is the subapical region of the cells. And this is how we make cells. This is how we outline all the cells. So remember, we label the Drosophila coherence, that is an adherent junctions.